Have you ever marveled at the wide variety of life on Earth? From flamingos to meerkats, from Venus flytraps to cuttlefish, there are so many different species running, flying, growing, and swimming around on our planet. And all of them, plant and animal alike, no matter how different they may look, have something important in common. One thing that allows them to thrive, evolve, and reproduce. One thing that contains enough variability to account for the wide variety of life all around us, yet enough consistency that its structure is instantly recognizable. DNA. Deoxyribonucleic acid, or DNA, is composed of 4 different nitrogen-containing bases: adenine, guanine, thymine, and cytosine. 2 of the bases, adenine and guanine, are purines, meaning they contain a double ring. The other 2, thymine and cytosine, have a single ring and are known as pyrimidines. Furthermore, as researcher Erwin Chargaff discovered, while each species has its own percentages of each base, in any organism, the amount of adenine always equals the amount of thymine, and the amount of guanine always equals the amount of cytosine. Put more simply, A equals T and G equals C in all forms of DNA. These 2 discoveries, that the amounts of each base varied from species to species and that in each species, A equals T and G equals C, have come to be known as Chargaff's rules. The DNA molecule is shaped like a twisted ladder. Deoxyribose sugar and phosphate groups in the DNA bond to form the sides of the ladder, while the nitrogenous bases make up the rungs. Adenine is always paired with thymine and guanine with cytosine. You could think of it as a dance where adenine is always facing thymine as its dance partner across the ladder, and cytosine is always partnered across from guanine. This is known as complementary base pairing, and it ensures that each rung of the ladder has a consistent width all along the length of the double helix, since adenine and thymine base paired together have the same width as guanine and cytosine paired together. In order for organisms to grow, repair tissue, and eventually produce, cells need to be able to divide and multiply. But before that can happen, each cell needs to be able to make an exact copy of its DNA. This copy can then be passed on to the daughter cells as division occurs. The first step in this process is the unwinding of the double helix. This is done with an enzyme called helicase, which breaks the hydrogen bonds between nucleotides. As the helix unwinds, new nucleotides will complementary base pair onto the now single-stranded parent strand. The new daughter strand is synthesized with an enzyme complex called DNA polymerase. Breaks in the sugar-phosphate backbone will be sealed by an enzyme called DNA ligase. DNA replication begins at numerous sites, known as origins of replication, all along the length of the chromosome. At each origin of replication, a replication fork forms, and around each fork, a replication bubble. DNA replication occurs inside these bubbles and moves in both directions along each strand until the process is complete. Replication of the diploid amount of DNA, over 3 billion base pairs in the human body, is completed in a matter of hours. The DNA of each cell contains all of the genetic instructions that determine the characteristics of that organism. That little twisted ladder is responsible for all the life you see around you. And it's what all that life has in common.